Okay, so students, I would like to first talk a little about the syllabus of minor. Um, just give me a second. I'm going to share it. So if you can see the sheet, yeah, in green is what we have already covered, okay, and in yellow is what we will be covering before the minor. So everything in green and yellow is going to be the syllabus for your minor exam. Today we will be talking about, we'll finishing principles of nutrition that we started in the last class and talk about bacterial reproduction, and then in the next 5 classes before the minor we're going to cover the other topics that you see here. So approximately 5.5 or 5/1/4 module will be over by minor 1. Now, after the minor and before the mid-sem break, we have, I think as far as I calculated, 9 lectures in which I would like to cover the remaining portions. Like you see, there's one viruses left here and these topics. I will finish those. I'll try to cover it. Because after the mid-sem break, there are 8 lectures, out of which I would like to use 5 or 6 of them for your Viva and one for the other quiz that is remaining. So that's how I've planned the course, and I just wanted to show you this so that you are not, it doesn't come to you as a surprise as to what the syllabus of minor is. Any questions regarding this? Okay, if not, then we'll come back to our lecture. Before that, can one of you tell me what was the assignment? What is the difference between SP dot and SPP dot?
Ma'am, it is SP dot is used when the name of the species needed to be specified, and SP dot is used when we are specifying many species within a particular genus.
Sorry, Devpriya, I couldn't understand. SPP dot is when you don't want to specify.
No, both are used when we don't want to specify the particular species, when it is not needed to specify. So SPP dot is used to refer to several species within a genus, and SP dot is used to refer to a single species within a genus.
तो वो तो सिंगुलर प्लूरल बोला ही था ना, वो तो खोलोगे जैसे ही विकिपीडिया पे आ जाएगा कुछ और एनीवे ठीक है चलो और कोई anybody else who saw what is the difference between SP dot and SPP dot? So this is true that SP dot is when you're referring to a particular species, but SPP dot is when you're talking about a group of species. But SP dot is also used when, for example, you're referring to a particular species, like you're writing the text and you say Escherichia coli or you say Pseudomonas fluorescens in the text earlier somewhere, and next time when you write you might just say Pseudomonas species, so you're referring to, then you know that it's that particular species you're referring to. तो ये भी एक यूज है. सिंगुलर प्लूरल तो तुम्हें सीधे विकिपीडिया पे मिल जाता कि एक स्पीशीज है और डबल पी डॉट है, लेकिन एसपी डॉट भी आप लिखते हो जब व्हेन यू डोंट हैव टू explicitly mention the species name, just say species, so people know that the one that you're referring to earlier. And that also reminds me that when you're writing, for example, S aureus, which you mean to refer to as Staphylococcus aureus, you have to see, and why I'm telling you all this is because I've already given you the term paper topics. Huh? So when you say S aureus, the last time that you would have described a generic name with S should have been Staphylococcus, because S aureus here is Staphylococcus aureus. Let's say the last genus name that you have mentioned in the text is Streptococcus, then you cannot write S aureus, because then it will mean that the generic name that you're talking of here designated as S is Streptococcus. समझ में आया? गोपिका समझ में आया?
No.
तो तुमने यूज किया, there are two names, let's say Streptococcus and Staphylococcus. ठीक है, there are two names of genera that you want to use in your text, and the species name that you would like to use somewhere down in the text is Staphylococcus aureus. Okay, so the last time that you would have used the genus name in expanded form should have been Staphylococcus, only then you can abbreviate it as S. aureus. Otherwise, it will be confused with the last name, that whatever you have used last in the text, it will be confused as that generic name. It represents that generic name. ठीक है, तो लास्ट टेक्स्ट में तुमने यूज किया होगा, अगर तुम फ्लो में जा रहे हो और तुमने यूज किया स्ट्रेप्टो स्टेफाइलो, 3 बार तुमने ये यूज कर लिया एसपी डॉट करके या जो भी, नाउ यू हैव टू यूज द स्पीशीज नेम, यू हैव टू चेक कि तुमने लास्ट नेम क्या जेनरा का यूज किया है, क्योंकि ये वही इंप्लीमेंट करेगा, इंप्लाई करेगा. अब क्लियर है गोपिका?
What's aureus?
Sorry, again Gopika? It's a
Aureus.
species name, Staphylococcus aureus. If I say that's the binomial nomenclature for Staphylococcus, Staphylococcus is the generic name. अगर तुमको Linnaeus वाला binomial system of nomenclature याद है तो Staphylococcus aureus is genus and species name. ठीक है? And I told you, I don't know if you've gone through that lecture. Anybody who has, Jaskeen, Bhavya, कोई किसी ने Carolus Linnaeus वाला nomenclature याद है binomial system? कोई मुझे बताएगा? देवप्रिया, विशंतन?
यस मैम.
मैंने बताया था ना कि जेनरिक नेम को आप अब्रीविएट कर सकते हो फर्स्ट कैपिटल लेटर से,
Yes,
है ना?
ma'am.
उसके लिए हम बता रहे हैं कि रैंडमली कुछ भी तुम एस से बहुत कुछ हो सकता है. तो एस ऑरियस, अगर स्टेफाइलोकोकस ऑरियस को अब्रीविएट करोगे एस ऑरियस की तरह, तो तुम्हें ये यू हैव टू मेक श्योर कि लास्ट वर्ड स्टेफाइलोकोकस तुमने यूज किया है टेक्स्ट में. अदरवाइज इट कुड बी एनी ऑफ द, इफ यू रेफर टू 2-3 जेनरिक नेम स्टार्टिंग विद एस, इट कुड मीन एनीथिंग. ठीक है, इतना क्लियर है भव्या?
Yes, ma'am.
Yes ma'am.
Okay, so this was about species SP dot and S double P dot. Let me start with what we did in the last class. I'll just share. So in the last class, we were talking about nutrition. Then we talked about what are macronutrients and micronutrients, or major elements and minor elements, or trace elements. And then we went on to discuss the uptake of nutrients, which will be, I guess it will be taken up in detail in your biochemistry class as well. But I wanted to tell you about the two modes of transport. One is the passive mode and the other one is the active mode. Passive mode is along the concentration gradient without the requirement of energy, while active would require energy. And passive may, I talked about simple diffusion, which is without the aid of a carrier protein, while facilitated diffusion requires a carrier protein, though even this is without the need of energy. And in active transport, I was talking about primary active transport, wherein at the site of transport, you have ATP being utilized, or whatever the energy source is. It could even be the proton gradient or any gradient that is generated. And one classic example was that of ABC transporter that I took. Then we talked about secondary active transport. So primary active transport is basically a uniport, while secondary active transport is a symport, which means two moieties can be transferred. And if the two moieties are transferred in the same direction, it's called a symport, while if it is in the opposite direction, it's called an antiport. Yahan tak we talked about it. And then I took an example of a gradient across the proton gradient across the plasma membrane, for example, H plus gradient, which would drive in the antiport fashion the transfer of Na plus, and Na plus can be transported back inside the cell together with sugar in the symport fashion. After that, I discussed about the third type of active transport, which is called as group translocation, because here you have a group which is translocated, so to the moiety that is being transferred. And the classic example here is of glucose being transported inside the cell, wherein during the process of transfer, you have glucose modified into glucose 6-phosphate using the P from high energy PEP, phosphoenol pyruvate. Okay, so after that, there are some nutrients wherein there are special mechanisms for transport. For example, iron uptake, if you see, iron is usually difficult because it's insoluble, and so it's very, it's rarely available to the cell. So what happens is several bacteria and fungi, they secrete what is called as siderophores. And siderophores are nothing but low molecular weight organic molecules that can complex with iron and supply it to the cell. So basically, once the iron siderophore complex reaches the cell surface, it binds to a receptor which is specific for siderophore, and then either the iron is released into the cell or the whole complex is then taken inside, let's say by the ABC transporter you just saw in the primary active transport. So, but this is specific for certain kinds of moieties. Okay, now there's a categorization or grouping of organism or classification of organism on the basis of nutritional types and just give me a second, please. Yeah. Based on the source of these three requirements, that is carbon and electron source, there is a classification that I'm going to talk about. Now, we have come across these terms during our earlier lectures. Some microorganisms can fix CO2, while others use organic matter as their source of carbon. So these are called as autotrophs, while organic are called as heterotrophs. Those that require organic carbon are called heterotrophs. On the basis of their requirement for energy, either they use light, then they're called as phototrophs, photosynthetic organisms, or they use energy from chemicals, and then they're called as chemotrophs. And the third requirement for a cell's growth is electron source, or for the metabolism also, you will see that the third requirement is electron source. This could be either from an inorganic source or from an organic source, and the terminology is lithotroph for inorganic and organotroph for organic. Now, if I've told you about these three terms, so auto hetero is for carbon, chemophoto is for energy, and litho organo is for, you can imagine types when you bring the three requirements together. So an organism, if you just categorize on the basis of energy source requiring light and chemicals, you can have chemo and photo, then either of the two could require either carbon dioxide or organic compound. So this goes on, right? There could be various kinds of organisms which can be classified. I mean, various combinations are possible. But the major nutritional type in prokaryotes are these four. And for you would not write it. So if, for example, there is photo, litho, autotroph. Now you can imagine from this, photo, litho, autotroph has light as energy source, litho as electron source, and it can fix carbon dioxide. Similarly, photo, organo, hetero, chemo, litho, auto, chemo, organo, hetero, but they are simplified as these are the four major groups. You simply say, so you miss out this electron source and you simply say photo, photo heterotroph, chemo heterotroph. मतलब लाइट एनर्जी का सोर्स और कार्बन के सोर्स को आप मेंशन करते हो. सो फोटो ऑटोट्रॉफ्स व्हिच इज लाइट, basically photosynthesizing those that can fix carbon dioxide, while light requiring but those which take energy from. So here I have even this table even has the source, and there are some representative organisms. But as and when we talk about different pathways in bacterial metabolism, I would like to connect you to what kind of can you hear me now, students? One of you, just keen.
Yes, ma'am, but it's breaking a little.
हाँ, but कहाँ से miss किया? मतलब it showed me bad connection, but from where? You got the when I was talking about these simplified terminologies, that was clear? One of you, Kashish.
Yes, ma'am.
ठीक है. So I was just mentioning that when we talk about metabolism, I'll try to specify that look, this is the chemical energy, or this is the source of electron that the organism requires. Here's just an example for you to know that there are representatives from each class that you find in organisms. Now, most of the non-photosynthetic bacteria, they are chemoheterotrophs. where the three requirements, if you see, are met by the same, either by the same or even different, but organic source. Okay. And these chemoheterotrophs, they depend, there are different ways in which it can obtain their organic nutrients. I think we have talked about this as well, but I would just like to specify here. Saprophytes are those bacteria which obtain their food from dead and organic, decaying organic matter, while parasitic are those bacteria that obtain their nutrition from the tissue of the hosts on which they grow. Okay, so this is from live tissue that they grow on. And symbiotic are those which live in close association with organism as symbionts, and they are beneficial also. So while saprophytic, parasitic would be negative to the host, saprophytic is of course on dead and decaying matter. Symbiotic is when they are in close association with a organism, and usually this is a beneficial interaction. So there is another class of organisms which they are called as mixotrophs, which is an organism that can use a mix of different sources of energy and carbon. So instead of having a single trophic mode, it has the ability to, let's say, shift from autotrophic mode to heterotrophic mode. Is widely spread in a variety of microorganisms, especially in prokaryotes and protists. So this is basically when they can switch modes depending on the conditions, and they're called as mixotrophs. And finally, I just wanted to present to you the trophic and energy pyramid, wherein this you might have studied somewhere in the school time. So if you look at this pyramid, starting from producers which utilize light energy, synthesize their own food, to primary consumer and secondary consumers, you will see just by the sheer number of microorganisms, the heterotrophs, so autotrophs are those that make their own food, heterotrophs are ones that depend on the autotroph or the producers. You will see that microorganisms, while they belong to all these trophic levels, the heterotrophs are majorly dominated by the prokaryotic microorganisms, just by the numbers they are present in. Yeah, so for microbial nutrition, I have just jotted down some reading material here. I would now like to move to the. Is there any doubt with nutrition, students? Because then I'll move to microbial growth.
Not really, ma'am.
So I had covered most of it in the last class about how. So अच्छा ये बताओ बायोकेमिस्ट्री में तुम लोग ने कर लिया क्या प्लाज्मा मेम्ब्रेन एंड ट्रांसपोर्ट अक्रॉस द मेम्ब्रेन?
नहीं मैम, अभी न्यूक्लिक एसिड्स एंड डीएनए आरएनए चल रहा है.
अच्छा ठीक है, वेरी गुड. हाँ, क्योंकि मुझे न्यूक्लिक एसिड के बारे में कुछ करना है. एनीवे ठीक है. ओके, सो we will now move to microbial growth. Microbial growth and nutrition happen to be the same module. Okay. We talked about the nutrients that are required by bacteria and how nutritional diversity is observed in this domain: photo, litho, photo, auto. I mean, basically photo, auto, photo, hetero, and stuff. These 4 domains. Now, nutrients are important for the growth of a cell. And upon achieving maturity, the cells would divide. So once they take up the nutrients, they would start accumulating biomass, and then once they achieve maturity, they would divide. So we will first talk about the cell division in bacteria, that is binary fission that you saw. I've told you earlier. And then we will, that when you talk about cell division, you're talking about individual cells and how they divide. And then we will look at how a population of cell grows. Before we do that, here is a recap of some cytoskeletal elements found in prokaryotes. Because they have a role to play in cell division. So I earlier mentioned just 3 of them: FTSZ, MREB, and chrysantin, if you remember. I want to draw your attention to FTSZ, which is involved in cell constriction during division, and MREB has a role to play in the shape of the cell, maintaining the shape of the cell. There are other elements which help in the segregation of plasmids. There are people, so Professor Preeti in the department works on plasmid segregation and the mechanistics involved. For us, what is of interest here is FTSZ and MREB. Now, the most common mode of cell division in bacteria is by binary fission. Binary fission matlab splitting into two. While this seems quite simple with the cell growing and then splitting, the mechanisms involved are being unraveled by scientists. Now, why is this important? Why are people working on this? Studying the mechanism of cell division in bacteria has relevance for biotechnologists because once this is understood, it can be manipulated or targeted by specific chemicals or antibiotics for controlling them. Now, other than binary fission, few bacteria, they undergo some unusual patterns of cell division. Example, some undergo multiple divisions to form such multiple cells which are released. So earlier they are in a sac and then they are released. Something similar to what you saw probably in the fungi and the protista, while others divide by budding. In fact, in some cases, you will also observe some asymmetrical divisions that take place that I have not depicted here. But we'll restrict ourselves to binary fission, how the cells divide by binary fission. Okay, so binary fission, like I said, is the most common mode of cell division. Conceptually, like I said, it's simple. A cell needs to grow and then it grows to twice its starting size and then it splits. But it's not as random as it might look. In fact, it's programmed even in the sense that to remain viable or functional, a bacterium must divide at the right time, in the right site. At the right site, you should have the cross wall and must provide each offspring with the complete set of genetic material so as to maintain the integrity. Now, before binary fission occurs, the cell must copy its genetic material and segregate these copies to opposite ends of the cells. In bacterial genetics, we will cover DNA replication. I'm glad that you've already done nucleic acids and DNA structure, so it will be easier for me to go over DNA replication. Now, once the DNA has replicated, it is followed by formation of the cross wall at the right position, which separates out into two daughter cells. The order and the timing of this process, the processes that are involved here are DNA replication, then DNA segregation, then selection of a site for laying the cross wall, and finally invagination with the synthesis of new wall. These all processes are tightly controlled. And a key component of this machinery is the FTSZ protein, which forms a ring that is called as the Z ring. So basically, they are protein monomers of the FTSZ which assemble into a ring-like structure, and this is called as the Z ring. Other components of the division are then recruited. Once the FTSZ ring is formed, then you have other components recruited at the ring, which forms the cross wall. Now, this machinery is positioned so that the division splits the cytoplasm without damaging the DNA in the process. So if the splitting takes place earlier, you will see that the DNA might get damaged. So there has to be a programmed synthesis or formation of the Z ring and then its constriction to form the cross wall. There currently exist several models and mechanisms that regulate Z ring formation, but the mechanisms depend on the species that we talk of. In fact, in a later slide, I'll also show you how FTSZ may not be present in a particular group also. Several rod-shaped bacteria like E. coli or even Colobacter crescentus, they use one or more inhibitor of FTSZ that forms a bipolar gradient in the cell, enhancing the polymerization of FTSZ only at the cell center. What I mean by bipolar gradient is, so I'm going to take the example of MinCDE, which is a set of proteins which helps in properly positioning the septum or the cross wall in the middle of the E. coli cell. So what happens is MinC is actually an inhibitor of FTSZ ring formation, and MinC oscillates between the poles repeatedly, thereby keeping the MinC gradient on a at the center at its lower average concentration. The higher concentrations are found at both the ends. So it, like I said, it keeps oscillating. Thereby, the possibility of formation of a septum is only possible at the center. This is where FTSZ would assemble because of lower concentration of MinC. Okay. And Min D and E are also associated with MinC. So MinC is not the only protein which helps, but its concentration is what regulates the formation of rings of the FTSZ only at the center, exactly at the center. किसी को कुछ पूछना है इसमें से? नहीं. Okay. MREB, now I told you the role of FTSZ. What happens is this is the one that forms the ring, and then to this attaches the divisome assembly, or this is the one which would then help in formation of the cross wall once the DNA has segregated. MREB is a bacterial protein which I said is analogous to actin found in the eukaryotic cell. Now, almost all the non-spherical bacteria, they rely on MREB to determine their shape. What MREB does is it polymerizes to form a helical network of this sort just under the cytoplasmic membrane, covering the entire length. So, MREB determines cell shape by assisting the position and the activity of enzymes which synthesize peptidoglycan. So you would see this is where it would initiate the synthesis of PG, thereby giving it a particular shape. And it also serves as a rigid filament under the cell membrane that exerts outward pressure to the cell. So basically this is how it plays a role in maintaining the cell shape. Now here is just a comparative of the kind of genetic material that the structure genetic material of prokaryotes together with some eukaryotes like protists and animal cells. Then how does the nuclear material divide? So in prokaryotes it would be binary fission with DNA replicating and moving to the two ends, while in protists you will see that the chromosomes attached to nuclear envelope and there is the formation of mitotic spindle. We are not going into the detail of meiotic and mitotic division. However, just to draw an analogy, in the case of prokaryotes, you have FTSZ proteins assembling to form a ring, while other cytoskeletal elements help the prokaryotes and the eukaryotes in forming the cleavage or the furrow. FTSZ subunits from the ring, what happens is there is progressive shortening of the ring by removal of individual subunits. So there's one school of thought which believes that once the FTSZ ring constricts, it draws towards the center, the cell membrane and the other layers of the cell envelope, so that the final division or cytokinesis can take place and the cell would divide into two daughter cells. But that's one hypothesis wherein FTSZ shortening is believed to be the reason for cytokinesis. Then, this is just to show you the typical localization pattern of some of the important proteins involved in cell division, FTSZ, FTSA and MREB in different kinds of bacteria. So, for example, in E. coli, you would see these three distributed across the cytoplasm in a cell which is in the pre-divisional state, while in the divisional state, this is how you would see the association. You see FTS Z become. I'm using these names so that you become aware of what they actually refer to, right? But again, okay, I'll show you an example. So this lacks MREB and FTSA. It just has FTSZ. While Staphylococcus aureus, which is it is a spherical cell. This lacks MREB. And the pathogen Chlamydia trachomatis. Now I'm specifying the name of the genera, otherwise it would have been confused with what I said here, Corynebacterium glutamicum. So in a cell or in your text, you cannot write C. glutamicum and C. trachomatis together, because then nobody will know what is C here, what is C referring to. That's why I specified this is Corynebacterium. I mean, not that I want you to remember here, I'm just trying to tell you how you write scientific names. And chlamydia, here the C is referring to chlamydia. So chlamydia trachomatis, this lacks both FTSZ and FTSA. In fact, here MREB has a role to play in cytokinesis. So this is just to show you that there's a range in the kind of skeletal elements which are involved in cytokinesis. So this was about the division of one cell. But looking at the larger perspective, when organisms are actually grown in closed systems or when they're grown in batch culture मतलब ये तो पढ़ लिया ना तुम लोगों ने, ये सब तो किया है ना, batch culture, closed system, anyone? Naman Singh? रुद्रांश चौधरी? कहाँ हो तुम लोग, म्यूट कर करके गायब? हाँ, कोंडापल्ली आश्रिता? वेरी गुड. कौशिक दास? ओके. दीपक कुमार जहांगीर?
Ma'am, in BBL 133, mass and energy balance, there are different type of process which is used to culture bacteria, and in which them there are some batch process, continuous process,
तो
and
बैच का
some
क्या
type of.
डिफरेंशिएटिंग फीचर है? व्हाट इज बैच कल्चर?
Ma'am, that I don't know.
मैंने तो खुद ही क्लोज सिस्टम और बैच कल्चर बोला ना. एनीबडी एल्स? जसकीन सिंह?
Ma'am, the way we grow bacteria and other microorganisms in the lab.
क्या मतलब है इसका? what does this mean? The way we grow मतलब we grow as in continuous mode also.
Ma'am, is it like
And
you
like
have
we give
bacteria that are provided like similar conditions, like similar nutrition, temperature, like etc.
नहीं
And
बाबा, तुम लोग क्या BBL 133 कर रहे हो? भव्या
and
बोलो.
l- with
बैच
less
में मतलब
location.
पहले जब एक पूरा हो उसके बाद दूसरा शुरू होगा।
हाँ, मतलब दूसरे की तो बात ही नहीं है. एक सेट में, एक क्लोज सिस्टम में आपने न्यूट्रिएंट्स डाल दिए. हाँ आशी बोलो, तुमको कुछ ऐड करना, बताओ.
मैम, इन द कंटिन्यूस प्रोसेस जो फ्लो होगा वो काफ़ी कांस्टेंट होगा. बैच में इट विल बी लाइक आफ्टर अ सर्टेन अमाउंट ऑफ टाइम यू गेट द प्रोडक्ट.
अरे नहीं यार, तुम लोग तो क्या बोल रहे हो मुझे? ध्वनि?
Ma'am, in batch process, maybe we provide a certain fixed amount of nutrients, and then while in continuous, there is maybe a continuous of nutrients. In batch process, the nutrient being used culture would decline, but in continuous it won't.
Yes, Dhvani. So I was expecting some more confidence, but with this course that BBL 133 में तो तुम लोग का पूरा batch और continuous से ही शुरू हुई होगी सारी की सारी तुम्हारे syllabus. I mean, batch is a closed system. So you put in all the nutrients in one go and let the reactions take place or let the organisms grow. While in continuous mode, there is a continuous feed of nutrients or any of this. It could be X amendment, and there's a continuous withdrawal as well. So batch is when, for example, you're growing organisms in a flask or in a tube. That's a batch culture. You give everything in one batch. Hence, it's called as a closed system. Lekin, from the responses that I've heard, In the next class you're going to explain it to me कि क्या होता है batch और क्या होता है continuous, है ना. Name itself says batch है, एक batch mode में वो grow कर रहा है. One set of nutrients or whatever amendments are required have been provided. And like Dhwani said, it has this what do you say? It has this disadvantage that it would the nutrients would expire, would exhaust at certain point of time. While in continuous, you try to see that there is a continuous inflow of the nutrients as well as an outflow, because you cannot just keep on adding nutrients without removing a similar volume. But again, I would like to know from you all, and please पढ़ के आइएगा जैसा बैच. और ये तो मुझे लगा था तुम लोग मुझे समझाओगे कि ये क्या होता है, तो यहाँ तुम लोग पता नहीं क्या क्या बोल रहे हो. जैसे हम लोग ग्रो करते हैं लैब में, और ये सब क्या बोल रहे हो तुम लोग मुझे. ये देख के जरा आप लोग आइएगा और मुझे नेक्स्ट टाइम बताइएगा कि बैच कल्चर और कंटिन्यूअस कल्चर में क्या डिफरेंस है. मैंने कहा भी कि बैच कल्चर और क्लोज सिस्टम. सो क्लोज सिस्टम मतलब देयर इज नथिंग दैट यू आर एडिंग or removing from the system while the growth is taking place. And why I'm talking about this is usually you grow organisms in the batch mode in the flask, and there what happens is, that is the assumption that we're taking here when we're talking about bacterial growth, when the population grows. Okay, so when you grow them in batch culture and the growth is plotted as log number of viable cells versus the time of incubation, the curve that is generated, ये curve तो पढ़ा होगा, ये curve पढ़ा है mass energy balance में, कोई मुझे बताएगा.
जी मैम, लाइक BBL 133 में बिल्कुल लाइक अलग स्ट्रक्चर है. इट्स नॉट रिलेटेड टू बैक्टीरिया और माइक्रोब्स एज सच.
Whatever the case भी, ठीक है, ये नहीं पढ़ा तुमने, लेकिन बैच और कंटिन्यूअस तो 100% पढ़ा हुआ है उसमें. वो तो
हाँ
स्टार्टिंग.
तो मैम लाइक वी कैन अंडरस्टैंड द कर्व.
ठीक है, ओके, परफेक्ट. वी कैन अंडरस्टैंड मतलब इसका क्या मतलब है?
लाइक द कर्व इज प्रीटी क्लियर टू अस. लाइक
दिस कर्व है ना. One of you.
मैम लाइक कैन आई ट्राई?
हाँ बोलो प्लीज.
Ma'am, like in the starting, you have like a fixed number of bacteria, and when you provide the nutrients, so they start, because in the starting the number of bacteria are pretty less, so they start growing exponentially because for them the nutrient availability is pretty high. So then that's why you plot the log number of cells, not the like net number of cells. Otherwise,
Direct
exponential
numbers.
curve aa jata.
Hmm.
And since it's logs, so that's why it's a straight line. And in the stationary phase, basically the like it reaches an equilibrium point where the amount of nutrients are just sufficient for the number of bacteria present in the culture. And after that, when they consume those nutrients, the nutrients starts like diminishing, and the bacteria start dying out.
Good.
So that's the death phase.
हाँ, so basically जब भी ये closed system है, ये continuous system में नहीं होगा, ठीक है. This is only when there is a closed system that you find these 4 distinct phases. And Vishantan, you very clearly correctly said that initially you observed this exponential phase, but you missed out on a lag phase, Vishantan. There's a very clear lag phase observed in bacteria in such a curve when you put in bacteria to grow in a medium. There's a distinct lag phase, which is basically because the cells are acclimatizing to the new conditions that you put the cell into, and then you observe this kind of an exponential phase wherein, because of the presence of these nutrients in abundance, you will have this kind of an increase. And then as nutrients start exhausting, you start observing this stationary phase, and finally there is a rapid cells. There is no fresh medium and everything. These are to a fresh medium which has rich source of nutrients. And in certain cases, or in most of the cases, you will see that the cell number does not reach 0. So they're not all killed. Every bacteria, like they have a mechanism to actually evade the stress. In this case, it's of nutrient deprivation. So you will usually also observe a 5th stage, which is called as a long-term stationary phase. But in most of the textbooks, you will just observe these 4 phases. Now, I'm going to talk about each of the phase and what happens in this phase physiologically and biochemically. What is it representing? Starting with the first phase, which is the lag phase. So basically, यहाँ पे क्या हुआ है? In a batch mode, you've added some cells provided them with nutrients so that they can grow. This is the incubation time. The cells that you've added, the initial sample that you've added is called as an inoculum. Okay. So what happens is, after a liquid culture is called as a broth, is inoculated, the multiplication of bacteria does not start immediately. And this time lag between the inoculation and the beginning of multiplication is known as lag phase. The lag phase of bacterial growth is important from various perspectives for a biotechnologist as well, but it is difficult to study because of low cell numbers, low cell density, and also because the metabolic rate of cells is quite low in this stage. In fact, it's actually believed that the lag phase, there are two phases of the lag as well. In lag 1, there is no accumulation of biomass. So it's not that the cell would immediately start growing and collecting biomass, accumulating biomass. But then the cell would start accumulating biomass, and then there's a phase called as lag 2, wherein there is accumulation of biomass, but there is no division. And only after this phase you will see that the exponential phase starts. So during lag 1, what happens is the ribosomal genes and the amino acid biosynthesis are shut down, preventing cell growth. It's only preparing the cell prime of the lag phase. This would even depend on the bacterial species. Then what is the composition of the culture medium? What is the growth condition? What was the history of the cell? Where did it come from, originate from? And in nature, what you see is that the bacteria, they adopt a lifestyle which is called as feasting and fasting. Okay, so they're always in this extremes. Bacteria spend most of their time in the stationary phase that you just saw due to limited nutrients in the natural environment. But what we are talking of is in vitro condition in the lab, wherein you're providing nutrients in excess. So in the stationary phase, what happens is the cell undergoes a number of adaptations when it is supposed to stay in the phase wherein nutrients are limiting. The PG layer thickens, the genetic material starts condensing, the ribosomes are temporarily deactivated as protein synthesis does not take place, and usually you also see a reduction in cytoplasmic volume. So a lot of biomolecules, they accumulate oxidative damage as well. With the kind of generations that have passed, they accumulate damage. So if the cells are to start dividing, all these steps that or all these changes that I just mentioned, they have to be reversed. So an important feature of the lag phase is to repair and to replace the damaged components. This is what I mean to say when I say it's preparing for the exponential phase. And when you inoculate bacteria, there can be two conditions which are identifiable. Either the cells would have been transferred to a poorer medium compared to where they were earlier. This is called as a shift down, or they have been introduced to a nutrient rich medium, in which case it's called as a shift up condition. So in either of the case, it has to prepare its machinery to start growing in that particular condition that it's now in. Now, once the cells have accumulated all that they need for growth and they have repaired their machinery, they proceed to cell division. In this phase, microorganisms grow at the maximum potential given the environmental conditions. Population is the most uniform in terms of chemical and physiological properties during this phase. In fact, this is the reason why most experiments, they use cells from the exponential phase for their particular, whatever assay they would like to consider. Balanced growth here refers to the cellular constituents being synthesized at a constant rate relative to each other, which in the earlier phase, that is in the lag phase, was unbalanced. So if a cell's generation time is 20 minutes, Generation time refers to the time taken for a population to double in number. So if it is 20 minutes, this is how the population is going to grow in the exponential phase. Cause it here for you, okay. The increase in population is always 2n, where n is the number of generations. So the final population nt, that is n after time t, will be n0 into 2n. And this is the kind of values that you get, which when you plot on, and Vishantan was telling about this, so if you plot on arithmetic scale and logarithmic scale, you will notice that the growth curve is exponential. You look at the log plot. Now, due to the predictability of growth in this phase, this is the phase which is used for calculating the time taken for bacteria to double in number, which I just mentioned as generation time. So we can describe the exponential growth, like I mentioned, as nt is equal to n0 into 2 to the power n, where n0 is the initial cell number, nt is the final cell number, and n is the number of generations. And now taking log of both sides and solving for n, we get n is equal to log nt minus log n0 over 0.301. The rate of growth during this, now again, you have to see that we're only talking about batch cultures. The rate of growth during this phase can be denoted in terms of the mean growth rate constant, which is k. And k here refers to the number of generation per unit time. Okay, so it is basically n over t, and plugging in the value of n from the last equation, the mean generation time. So if you see, it will be nt minus log n0 over 0.301 t, and the mean generation time or the mean doubling time can now be calculated. If the population doubles, t will be g. So nt is equal to 2 and 0, and you substitute 2 and 0 in this equation. What you get is k is equal to 1 over g. That is, the mean generation time is the reciprocal of the mean growth rate constant. And another, you can even directly determine the generation time by a microbial growth curve, wherein population data is plotted on log axis for number of cells. Then the time to double the population is directly from the plot. This is, however, a value which differs from organism to organism, and it also depends on the environmental conditions. You can understand that this is going to be, the generation time is actually going to be longer in nature compared to the lab conditions. Here are just some examples of different bacteria with their respective generation time. What you have to note is, whenever such kind of a generation time is being specified, you also specify the medium in which it has been reported, as the medium also plays a role in the generation time that an organism will have. Now, coming to the third phase, which is the stationary phase, with the exponential rise in the number of cells in the phase that we just talked of, and the system being a closed system, the nutrients start exhausting. So is the oxygen in start with the rapidly metabolizing cells synthesizing it. So the cells enter into a stationary phase, called so because here the rate of bacterial cell growth is equal to the death rate. So a net increase in the cell number is not observed. Now here there are a number of changes that occur in the cells in terms of their physiology as they adapt to the new starvation conditions. I already mentioned to you when I was talking about the lag phase that the conditions need to be reversed, and what were the conditions. So till now they had an abundance of nutrients. Now this is stress. Okay, they're limiting nutrients, so there's stress. The few new cells that are produced in the stationary phase are smaller in size. So for example, bacilli even becomes spherical in shape. Their plasma membrane, it starts exhibiting more hydrophobic molecules on the surface, promoting aggregation. Then the DNA to prevent damage, what happens is it starts becoming bound to special proteins called as DNA binding proteins from starved cell. They're called as DPS, DNA binding protein from starved cell. So the DNA is also to be protected. All these changes are to facilitate the cells to survive for a longer period of time under adverse conditions. And in nature also, these strategies are used by cells in the oligotrophic. Oligotrophic condition is referred to as a low nutrient environment. So similar kind of changes of the cell shrinking, of the cell clumping together, preventing its DNA forming thick PG layer that I already mentioned. These are some of the changes which you observe in the oligotrophic environment. And it's believed that cells in the natural environment, they typically exist for long period of time in oligotrophic environment after they have undergone these kinds of changes. Actually, in nature, it is only for brief periods of time that there is influx of nutrients that return them to the exponential growth. Otherwise, they're always in, or mostly in stationary phase. Now, this phase is important even for biotechnologists because during the stationary phase, the cells produce secondary metabolites, or the metabolites produced after the active growth. These are called as secondary metabolites. And many of these are of economic importance to humans. For example, even antibiotics are secondary metabolites of microbes. Now, in this phase, as you can also understand, that those cells which can form endospores, they activate the necessary machinery during this stage to enter into the dormancy phase. And I'll just cover up the next phase also before we end. The final stage, that's the fourth stage that we saw in the curve, was the death or the decline phase. This is where the death rate is higher than the growth rate of bacteria. It is thought that the culture conditions have deteriorated to a point where the cells are irreparably harmed. Since the cells collected from this phase, they fail to show growth when transferred to a fresh medium. It's important to note that if the turbidity of a culture is being measured to determine cell density or trying to see growth, then the measurements might not decrease. It is not that you see any decrease in turbidity, since cells would still be intact in this phase. Cells thought to be dead might be revived under certain conditions, and there's a condition into, or a phase into which some cells enter, which is called as viable but non-culturable. So you cannot culture them, but they are still viable. In fact, one of the term paper topics is VBNCs. This state might be of importance for pathogens where they enter a state of very low metabolism and they lack cellular division, only to resume growth at a later time when conditions improve. It's also been shown that 100% cell death, like I said, is unlikely for any cell population, as the cells, they mutate to adapt to the environmental conditions, however harsh the conditions might be. Often there is a tailing effect observed where a small population of the cells cannot be killed off, and hence this fifth stage that I was saying, the fifth stage that was not depicted in the growth curve. In addition, these cells might benefit from the death of their fellow cells. So this is there. I actually said that this is all programmed. The cycle, the life cycle of a bacteria is quite programmed. So the death of one would actually be beneficial for the others. So this death provides nutrients to the environment as they lyse, and they release their cellular content, which might be taken up by other bacteria, other living bacteria. Yeah, so I've talked about all the phases. I'll now move to, in the next class, I'll move to the factors which affect growth. I would like to take questions if there are any from what I've just covered. So we did bacterial reproduction, that is division of a single cell. And then we saw how the population grows, the four phases basically of bacterial growth in the case of a batch culture. And you will tell me what is a batch culture and what is a continuous culture in the next class. But as of now, anything that you have a doubt, please let me know now.
Ma'am, what exactly is the VBNC stage like? I didn't get it. Like,
तो VBNC का full form क्या था?
viable but not culturable.
Viable but not culturable. If in that state, so what happens is, like I said, there are different strategies for bacteria to adapt to any kind of stress, and VBNC is believed to be one such strategy wherein the cell actually goes into a state of dormancy. It's not forming a spore, but it goes into a state wherein the metabolic activity is minimized, okay, and they do not divide, but they are not dead. They are still alive. Under very specific triggering conditions, they can resuscitate. उसको resuscitate कहते हैं, germinate. जैसे spores तो germinate करते हैं, आप VBNC को कहते हो कि आप वो resuscitate करते हैं. और इसमें बहुत clear, you can even see the cell physiology क्या है. अगर आप इस not observe growth at all, ठीक है, but it does not mean that they are dead. They are non-culturable, so they need some triggering factors. ठीक है.
Yes, ma'am.
और अगर तुम्हें और डिटेल चाहिए तो तुम्हें इवन आई कैन पास ऑन सम पेपर्स व्हेन दे टेल यू हाउ डिफरेंट द सेल्स. सो बेसिकली ये होता है कि वाटर श्रिंकेज हो जाता है, लो मेटाबॉलिक एक्टिविटी चाहिए, वो नॉर्मल मेटाबॉलिज्म में है नहीं, ठीक है, तो उसको न्यूट्रिएंट्स भी उतने नहीं चाहिए, बट इट्स स्टिल सर्वाइविंग. ठीक. And this is just one of the type of cell. I mean, you can have, in fact, when I was talking about biofilms, there are other phases also, like persisters बन जाते हैं. VBNC तो बस एक phase मैंने तुम्हें बताया, बहुत सारे जैसे spores बन रहे हैं जिसमें कि एक बहुत demarcated cell envelope है. VBNCs में ऐसा तुम्हें envelope, thickened envelope नहीं मिलेगा, लेकिन the cell's physiology undergoes a drastic change such that even under low metabolic activity, the cells are alive. ठीक है.
Okay.
Okay, students, then if there are no, हाँ पूछो कशिश.
Yes, ma'am. Ma'am, in the session phase, ma'am, does the rate of reproduction is equal to rate of death, or the cells stop reproduction?
Sorry, प्लीज फिर से बोलना कशिश. इन द डेथ फेज, कौन सा फेज पूछा?
Stationary phase.
In the stationary phase, it is equal. So there is bacteria dividing as well. But death phase takes up. हाँ.
Ma'am, means some bacteria are reproducing while some bacteria are dying. Means the bacteria which are reproducing must be getting the sufficient nutrients.
अब एक मिनट, सबसे पहले तुमने बैक्टीरियाज बोल दिया कशिश.
Yes, I'm sorry.
है ना. हाँ, यस. तो इनफैक्ट जो डेथ हो रही है ना, वो ही प्रोवाइड भी कर रहा है न्यूट्रिएंट्स उनको, उस फ्रैक्शन को जो अलाइव हैं, जो ग्रो कर रहे हैं. ठीक है. इन उसके बाद ये कंडीशन आती है कि न्यूट्रिएंट्स इतना एग्जॉस्ट हो जाते हैं कि वो डेथ फेज में चल जाते हैं. स्टेशनरी फेज में देयर इज अ बैलेंस बिटवीन द ग्रोथ रेट एंड द डेथ रेट. तभी तुम्हें वो स्टेशनरी दिख रहा है. मैंने कभी नहीं कहा कि सेल्स डोंट डिवाइड और दे आर किल्ड. इवन इन डेथ फेज यू विल हैव सम cells, few cells which are growing. ठीक है, but it's just that the death rate is much higher, hence you see this exponential decline.
So ma'am, does that mean that in the stationary phase, the bacteria, because there is nutrients?
ऑक्सीजन की अवेलेबिलिटी कम हो रहा है. ऑक्सीजन को अगर न्यूट्रिएंट कहते हो तो यस, द न्यूट्रिएंट्स. देन टॉक्सिक्स बहुत अक्यूमुलेट हो जाते हैं, जो कि इनफैक्ट हम लोग टॉक्सिक्स कुछ रिएक्टिव ऑक्सीजन स्पीशीज और कुछ डैमेज की हम लोग इसके बाद बात करेंगे. So because it is a closed system. Had it been an open system wherein you are removing these nutrients and replenishing with fresher nutrients, you will kind of see the exponential growth continuing, right? But because there are also toxics accumulating, the cells will not have a favorable condition for growth. ठीक है ध्वनि?
Yeah, okay. Actually, I had gotten confused. I thought that maybe they were dying because of their end of their life cycle and not because the lack of
तो
nutrients.
वैसे तो तुम बैक्टीरिया को इमॉर्टल कहते हो if given. मतलब ये कहते हैं ना कि omni present and immortal and stuff, because one has to one divides into two, and if the conditions are such that they can proliferate, है ना. So that original mother cell has been dividing into two and then so on and so forth. So because the conditions are not favorable, it is no longer able to divide. Okay.
Yeah, okay.
Okay, so If there are no further questions, then we will stop here. We'll meet on Thursday, and they will take up the growth. We'll try to finish microbial growth in the next class. Okay. Okay, students. Bye bye.
Thank you, ma'am.
Thank you.